The recent research conducted at Columbia University suggests a future where parents might be able to repair disease-causing mutations in embryos before a child is born, potentially eliminating hereditary conditions like blindness, heart disease, and sickle cell anemia. However, this ability also raises significant ethical concerns about eugenics, which is the controversial practice of selecting or modifying desired human traits. Detailed research overview. Led by Dieter Egli, a geneticist at Columbia University, the team has utilized a new technology called base editing, which is a method that allows scientists to meticulously replace incorrect genetic letters in DNA sequences without the damaging side effects often caused by older methods. This marks a significant technical evolution from CRISPR, which is an older gene editing tool that creates customized molecules to snip out or cut a targeted segment of DNA. While CRISPR became a standard tool for scientists after its invention in 2012, it was not perfect. In some cases, the molecules failed to find their targets or caused catastrophic consequences, such as destroying entire chromosomes. The Food and Drug Administration (FDA) did, however, approve a CRISPR-based treatment for sickle cell anemia in 2023. Historical context and controversy. The field has been marked by controversy, most notably in 2018 when a Chinese scientist, He Jiankui, used CRISPR to alter the DNA of human embryos to provide resistance to HIV. His work was widely condemned by experts as reckless, and he was subsequently imprisoned by Chinese authorities for 3 years. The recent experiment. In 2025, David Liu, a geneticist at Harvard University, and his colleagues combined CRISPR molecules with other compounds to create the base editing method, which is considered superior because it is faster and does not involve cutting DNA strands. Dieter Egli and his team at Columbia University decided to test this new method on human embryos. They aimed to alter 2 specific genes. The EYS gene, which is associated with hereditary blindness, the PCSK9 gene, which can carry mutations that raise LDL, low-density lipoprotein cholesterol levels in the blood, and increase the risk of heart disease, the HBB gene, which directs the production of hemoglobin, a protein in red blood cells that carries oxygen in tissues. In their experiments, the researchers delivered these base editors into fertilized eggs and 2-cell embryos, and successfully avoided the massive genetic damage associated with earlier CRISPR attempts. Current status. Despite the successful results, Dieter Egli emphasized that this research is currently under review for publication in a scientific journal, and is not yet ready to be used in clinics. He called for a broader public conversation regarding the pros and cons of altering human DNA, noting that as a scientist his role is to provide the data, but society must decide how to proceed.